My name is Ana, and thank you. I want to thank the organizers, especially Gabriela and Maribel, for the opportunity to be here, to be back to Cuatro Ciénegas, which makes me really happy. And it also makes me really happy to share the work I'm doing now in the DIM team, in the University of Paisaje with David Moreira and Purificación López García. And I don't need to tell you that we live in a microbe world, and that everywhere we look we find microbes and ecosystems that are dominated by microbes, and that these microbes are responsible for cycling the elements on the planet and make life as we know it today. So this microbial world harbors many ecosystems, and life abounds in this mesophile. I'll take the pointer. In these mesophile ecosystems, where temperature is around 37, let's say, neutral pH. Doesn't work. I can use my pointer. It works? No. Okay. Neutral pH and salinity 0, and an atmosphere of 1. Well, but microbes have reached. Do you have the la antena del coche? The high-tech pointer? No, it's fine.
Oye, ¿quieres estar al día? Aquí sí lo hace. Todavía más analógico.
Well, it's not a problem, it's fine. Well, so, yeah, in the meantime, this is super high-tech. So, but microbes can conquer niches that others cannot. So they can inhabit temperatures of water below 0 or above the boiling point of water, very acidic pHs, very alkaline pHs, very high salinities, up to 10 times more than of our oceans. But salinity is a tricky parameter because it's not always about sodium chloride salts, so they can be magnesium salts and they can be calcium salts, and this affects the chaotrophicity and the water activity. That is how the water molecules can enter are bioavailable for the cell. And for this reason, we know only 3 lineages that have adapted to very high salinity, which is Salinibacter in bacteria, and the archaea and the nanoarchaea. And another question that we have is which microbes can adapt to not only to one extreme condition, but to many, to polyextreme conditions. So I'm going to talk about this place in the North Danakil Depression in Ethiopia. Here it lies the Dalol Dome in the mix of 3 tectonic plates. And the Dalol Dome is a protovolcano that is lifting 40 meters, and it's in this Danakil Depression that it's an ancient arm of the Red Sea, and it's very geothermally active, and this is all a salt crust of 1 kilometer depth of salt. So this is a unique site that combines different physicochemical parameters of pools with very acidic, hypersaline with magnesium-rich and calcium-rich ponds, and high temperature. So yes, life on Earth has adapted to all of these parameters, but do we see microorganisms that have adapted simultaneously to these conditions? So in 3 expeditions in 2016, 17, and 19, the team went to sample these different sites in the Danakil area, and I'm just going to talk you very quickly through the sites. So this is the Dalol dome. These are the hypersalid hot ponds. The pHs are negative from -0.2 to -1. The temperatures can go up to 108 degrees, and the salinity can go up to 45%. The water activity is not that bad. So the limit of life for water activity is around 0.7. And this is the Yellow Lake. The pH is 1.8. The salinity goes up to 40. The temperature goes up to 43 degrees Celsius. The salinity can go up to 60%, and the water activity is very low. And as you can see, it's bubbling gases, which are toxic, and you can see that they're highly toxic because there are dead birds around the lake. And this is the Black Lake. The pH is 3, the temperature is 70 degrees, and the salinity is 70%. This is only liquid because it's at this temperature, so if you take like a bottle and you let it cool down, it just solidifies. And the water activity is very low. Yeah. There are some other systems that are not as extreme. So these are the Western Canyon lakes. The pH is acidic, up to 5. The temperature is more mild, and salinity can go up to 41%. But the water activity is still low. This is a cave reservoir in the Dalol canyons, where the pH is 6, the temperature is mild, but the salinity is still 33%, and the water activity is fine. So we're now reaching more friendly environments of pH 6, temperature 26, and salinity 37. So this is a great, this is a natural laboratory of gradients of polyextreme environments. So we go from the Dalol dome, which are these very acidic pools, to the Yellow Lake, the Black Lake, and then we have this more friendly in this gradient, which will be the Western Canyon Lake, the Lagarot, the cave reservoir, and the Asali Lake. So, sorry. The work of Jody Relillas, she did the PhD thesis, and just to tell you, like very quickly, all of her PhD, she did a lot of approaches. So she did microscopy, she did metabarcoding, she tried to detect DNA via flow cytometry, and she did the enrichments. There is no life in these 3 sites. There is no life in the Black Lake, in the Yellow Lake, and in the Dalol pools. But there is life in these sites that are a bit more friendly. And she had a lot of controls, and she had a lot of care, but it's something that we need to take very cautiously, because we think of liquid water in the surface of a planet as a criterion for habitability, and maybe we should reconsider. So I told you in the beginning that this is a microbe world, and I on purpose placed these 3 images where there is no life. There is no life here, and the reason is the physicochemical parameters. So in this PCA, you can see the water activity, the sites for the water activity, salinity, temperature, and pH, and we can draw the life limits here in the presence of very high water, liquid water with very high calantropicity and low water activity, because these sites are dominated by magnesium and calcium salts. Okay, but I'm a bioinformatician, so I need life to work. So I'm going to tell you now what kind of life is there when we can detect it by approaching to these limits of life. Oops. So it's over. There's no life. Sorry. I don't know how to use this mouse. Okay. So, ya está, ¿no? Okay, so we need 5 metagenomes for these 5 sites, 2 Western Canyon lakes, 2 sites in the Asali Lake and the cave reservoir, and life is overwhelmingly dominated by archaea. So the sites that are more extreme, that are the Western Canyon lakes, it's basically archaea. It's 99% of the community is archaea and nano-archaea. And as you approach the more friendly environments, let's say, you start to see Salinibacter, which is the lineage that I told you that we commonly see as a halophile. But we also have some cyanobacteria in Azale Lake, and some which the cyanobacteria are absent from the other sites. Sorry, the Viking. And but we see some lineages that we didn't know they could be hyper halophiles, and they are here. And so just a quick reminder, so these places are dominated by Halarchaea and Nanohalarchaea. And Nanohalarchaea are members of the Dipan superphylum, which are parasites or epibionts of other archaea, particularly the Nanohalarchaea. So these Halarchaea and Nanohalarchaea are probably in a type of mutualism or parasitic or symbiotic relation. So, okay, so how is life adapting to this very salty and acidic and extreme environment? So one of the adaptations for very high salinity is to look into the PI, the isoelectric point, the composition of the proteomes. So here I have a gradient of metagenomes that of different salinities. So it goes from fresh water to seawater in the Mediterranean, and from different solar ponds in Alicante that go from 6 to 14 to 32% salinity, and then we have the Dalol sites. And in normal, like fresh water and marine water, the PI profiles of the proteomes normally follow a bimodal distribution, with one peak in proteins being acidic and one peak in proteins being basic. But as you start shifting towards higher salinity, the profile of these proteins, they start shifting towards a very acidic profile of the proteins. And here you can see that the Western Cairo lakes encode the most acidic profiled proteins ever observed before. So we also reconstructed genomes out of the metagenomes, and we have like 160 high-quality mags, mostly from Halarchaea and Dipan. These are mostly nanohalos, because that's what we have. We have also some 30 high-quality mags from bacteria. And these include novel lineages of halophiles. So we have novel Halarchaea that are basal for the rest of the Halarchaea. And we also have a novel class, I think, a novel family of Nanohalarchaea. And we name these we use words that mean salt for the Afar people that have been living here for millennia extracting the salt. And so we named the Dipan the Aswanarchaea and the Halarchaea, the Afararchaea, and the Chuarchaea, which are related to salt. And Brittany Baker, her PhD project was related to using these mags, and she used them to increase the sampling and to resolve the tree of archaea. And she found that in the archaeal life history, archaea had adapted to hypersaline environments in 4 independent occasions. And so archaea are pushing these limits of adaptation in these sites. And here we have a BCA of their genomic features. So these are, on this side of the plot, you have more more basic amino acids, and you see that the metagenomes that fall here are the ones from fresh water and the Mediterranean, and the southern ports that are not so salty, like 4% and 16%. And here on the side of the acidic amino acids, you have the metagenomes from the Danakil depression, and the salar pond that is at 33%. And so, well, I don't know if I have time to go into why these amino acids, but we can discuss that. And if you look into the mags, you see that the PA profile of each of the mags, so the archaea keep this very acidic proteome that matches a salting strategy, while the bacteria not so much, and these gray points here are the CPR, which follow a completely different trend. And if you divide the mags by site, you can also see that the PI also decreases along with the gradient of salinity. Also, we think, or it's been said a lot that as the environments become more extreme, the diversity lowers. So here I'm showing you the diversity indices for the metagenomes. And in green I show all taxa, and in the brick color I show the hierarchia, and the pink the nanohierarchia. And you can see that this is again my metagenomes from freshwater, seawater, and here are the solar saltants. And here you can see that, yes, these trends follow. So as you for all taxa, as you approach the gradient of salinity, diversity decreases. But you can see in the southern ponds that the diversity of the hierarchia and the nanohierarchia start to increase with the gradient of salinity, and you can see that this explodes with in these lakes of Dalol, where we have within the hierarchia and within the nanohalo we have hyper diversity of these lineages. We also have a new lineage of a new Marchiota that is within the Dipal superphylum. And there were already some mags in GTDB, so I took those mags and our 3 mags from the Danakil Depression, which are depicted here in red, and you can see that they are also following this salting strategy, and they have this acidic proteome. So this is a new clade of hypersaline archaea. So this represents yet another, a 5th adaptation of archaea to extreme allophily. And you can see that this was acquired because the rest of the mags from GTDB, they don't follow this trend in their genomes, so they don't have this feature. We also have a new phylum of bacteria that we named the Satsura bacteriota. And there were only 2 mags in the GTDB, so we are contributing with 3 more mags. And the thing that I find super interesting here is that they have probably adapted to this extreme halophily due to horizontal gene transfer from archaea. So with this, I started my talk telling you that we have 3 main lineages of or microorganisms that have adapted to hypersaline environments, which were the Salinibacter, the Haloarchaea, and the Nanoarchaea. But here we're expanding to 2 more lineages, the Halonic Marchiota and the Satsura bacteriota. And just to show you how these organisms are living, or because I told you at the beginning that there are no cyanobacteria and there are also no phototrophs. So this is mainly dominated by heterotrophs, and they are relying on heterotrophic processes. And they are mostly degrading amino acid and fatty acid, and they're utilizing carbon. So these places are bubbling with water bubbling with gases in the deep, and they are following a feast-famine strategy. So when the gases bubble from below, they can feast, and they can feed on whatever hydrocarbons are bubbling, and they utilize it. But when the desiccation time comes, they store this carbon because they have carbon storage genes, such as the polyextreme biosynthetic genes. And just very quickly, I will tell you that we are also studying the viruses in collaboration with Mart and Yifan in the Pasteur Institute. And they have identified new viral families, and they have cultivated them. And they identify the host via the CRISPR spaces. And what is super cool about this is that it's just adding to the layer of complexity. So we have the alloarchaea that are being probably parasite. Well, I didn't talk about it, but they're probably more in a mutualistic relation, because the nanoalbarchaea have genes. They have amylolytic enzymes that could be helping the albarchaea to maintain these stable relationships, and that's why we see that the community is always albarchaea and nanoalbarchaea. And but there are a lot of viruses that are attacking both of them, and these viruses they share genes via horizontal gene transfer between them, but the viruses for albarchaea cannot infect the nanoallos and vice versa. So the viruses have diverged, even though they share genes via horizontal gene transfer, they have diverged. And there's a lot of these spindle-shaped viruses that they don't have now a taxonomic classification, but probably they will become a viral phylum of their own. And these have virus satellites. So these viruses have virus parasites that are being that are parasitizing the viruses with the virus enters into the cell to replicate. So yeah. So the take-home messages is, well, first of all, that liquid water doesn't mean that life is there. We need to remember that, and because of the presence of the salts, the water can have high chaotropicity and low water activity. But in these polyextreme lakes where life is possible, they are overwhelmingly dominated by archaea. And we have expanded the known diversity of extreme halophiles, and this high diversification of extremophilic archaea in geothermal influenced brines, and we have identified new archaeal families and a new phylum of halophilic bacteria. Their adaptation is via very extremely acidic proteins, and they have a high metabolic flexibility following these feast and famine strategies. And I want to thank my team, the team led by Puri and David, and our collaborators, and you for your attention.
We have a little time for some questions, if you want. Very late.
I'll be around, so we can also.
Some comments or final reflections, because this was really a great session about. Yes,
I never thought I would hear a limit of life. I thought that life would conquer everything, but it is amazing, the black pool, and that there's, forget Atacama you put some water and everything is alive. But these places are really, really amazing. Do you think there's more places like this, or is just this particular spot in life?
We think there is one more place like this in the Atacama Desert, but we need to dig deeper. Also, the difficulty of this place is that it's very hard to reach. So Dalol, the Danakil Depression is in the border with Eritrea, and they're in war. So it's hard to go back, and it's hard to take samples. So for some of the sites, sample there, you need to reach them by helicopter. So the access is very difficult. Well, not to the Dalol ponds, because those are even touristic, but yeah, it's not easy to get there.
That is a great works. I have two questions. One is how you took the samples and protect yourself from these very dangerous spots.
Well, I wasn't there. I was not fortunate to go. I arrived to the lab where they have already taken the samples, but they use gas masks. And well, they go with the AFAR people, so they know how to reach the places, where to walk, where not to walk.
That could be a really nice.
Yeah.
And the other question is, do you have? Well, I don't know if you are part of this culture, archaea culture, but they have anoxic protocols to culture them? Or?
So these are oxic environments. So these are not anaerobes. Archaea are normally aerobes. We tried to culture them, and we succeeded in some, but not in the new. We couldn't culture the ones that their new families. We could culture the most normal Archaea.
Thank you.
Mariano, please.
Thank you very much for the presentation. It's amazing. Do you know or have an idea what is the reason of the acidity of this water? Why is this process of should be a volcanic process that is?
Well, I'm not the expert, but I'm going to try to explain it, but I might tell you something that is not very. So there is a crust of 1 kilometer of salt, and this is a volcano that is forming. So I'm just gonna mess it up. No, I think I'm gonna pass on answering this question, but I can refer you to this paper.
Thank you.
I don't want to mess this up.
Really nice work. I really enjoyed your presentation. You're clearly in favor of naming taxa when there is no cultural representative, and I'm not against it. I think it's you found it, you discovered it, it's okay, you can. But what will happen when somebody cultures one representative and names it another name, and then it would break the code because.
So we registered them in SEC code, which they are trying to. I mean, if you don't name it, then the name that they will have is XJ43. So we're trying to give them names that reflect where they come from or something about their metabolism, but keeping in mind that as we sequence more, or as we discover more, or as we culture more, the lineage, the sisters of those could have some differences. So we try to keep this in mind, and we also try to retribute, like to do like a bit of a tribute to the place where we sample. But these are guidelines that are set by the GTDB and the SEC code, and we are following them.
Okay, so there is going to be two distinct codes, one microbiological and the other one, the one that's SEC.
SEC code, they're trying to but microbial taxonomy is a bit of a mess. So I'm really fond of. I know it's not perfect, but I like what the people from GTDB are doing because they're trying to set some standards based on the branches of the trees to set up the limits. Of course, as we sequence more, the branches change, and we redefine limits, and it's messy. Yes, but as Karen said, that's the best we have, I think.
Thank you.
Thank you. A last one? Or a last reflection?
Thank you for a very nice work. In those places where there is no life, has somebody trying to see which microbes grows closer to those places?
So we did several things. One of the things we set the traps for wind dispersal. But the thing is that it's mostly like, it's not like this sediment. It's like, it's like just salt crusts. I don't know, like maybe here you can see. And like this is what is next to. So it's just the salt that is left from the evaporation. And these are not always sodium chloride salts, or these are magnesium or calcium salts, so it's very hard to get DNA or anything out of there.